7%. That's quite a chunk of our genetic code, isn't it?
It really is surprising when you think about it.
All dedicated to building membrane transporters and related proteins.
These tiny gatekeepers.
So what are they actually doing inside us? Especially, when it comes to medicines we take.
That's exactly what we're going to dive into. It's this fascinating world of membrane transporters and their absolutely crucial role in how our bodies handle drugs.
Right. Not just if a drug works, but side effects too.
Exactly. Both the good and the bad, potentially. We've been looking at the scientific literature, really digging into this.
Okay, so what have the sources shown us? What are we looking at today?
We're looking at their different types. The main families are called ABC and SLC. We'll get into what those mean.
ABC and SLC.
Got it. And how they work. Some actively use energy, others are more passive, and importantly, where they are in the body.
And for you listening, the goal here is to understand how these transporters act like, gatekeepers.
Right. They influence how drugs get absorbed, where they go, how effective they are, and even things like drug resistance.
And it gets personal, right? Because our individual genetics play a role.
Definitely. Your specific transporter genes can really impact how you react to certain medications.
Okay. So our mission today, pull out the key insights, make this complex topic understandable and relevant for you.
Let's do it.
All right, let's start at the beginning. What are membrane transporters?
Well, fundamentally, they're proteins. They sit in the cell membrane, that outer layer of pretty much all living organisms, bacteria, humans, everything.
And their job?
Control traffic, what comes in, what goes out, nutrients, ions coming in, waste, toxins going out.
And drugs?
And drugs, absolutely, both coming in and crucially going out, too.
So like cellular border control.
That's a good way to put it. They manage the flow across membranes. For drugs, the two really big groups are the ABC, that stands for ATP binding cassette, and the SLC, or salute carrier transporters.
Okay, ABC and SLC. Let's take ABC first. What's their defining feature?
The key thing for most ABC transporters is that they're primary active transporters.
Active meaning energy.
Yes, they directly burn ATP, the cell's energy currency, to power their work.
So they can force things across, even against the natural flow.
Exactly. They can pump substances uphill against their concentration gradient. It takes energy, and ATP provides it.
Interesting. Any famous examples in this ABC family?
Oh, definitely. P-glycoprotein, or PGP, is a big one. Its gene is ABCB1, sometimes called MDR1.
PGP, I've heard of that. Involved in drug resistance.
Very much so. It's notorious for pumping chemotherapy drugs out of cancer cells, making them resistant. It's a major challenge.
Wow. Okay. Any others?
Another important one, though its function is a bit different, is CFTR, the cystic fibrosis transporter from the ABCC7 gene. It mainly moves chloride ions, but it's still an ABC transporter using ATP.
Right, linked to cystic fibrosis. Okay, so ABCs are the ATP-powered pumps. What about the other big group, the SLCs?
The SLC superfamily is much larger and more diverse in how it works. It includes facilitated transporters and secondary active transporters.
Okay, break that down. Facilitated.
Facilitated transporters help molecules move across the membrane down their natural gradient, so high concentration to low. They don't directly use energy. They just provide a pathway, like a specific doorway.
More like a revolving door than a pump?
Good analogy. And then secondary active transporters are a bit more complex.
How so?
They do move things against their gradient, like active transport, but they don't burn ATP directly. Instead, they harness energy that's already stored in an existing ion gradient, often a sodium gradient set up by other pumps, like the sodium-potassium pump.
Oh, so they're like borrowing energy,
What?
hitching a ride on another gradient.
Exactly. They use the downhill flow of one substance, like sodium, to drive the uphill movement of their cargo.
Clever. And you said this SLC group is bigger.
Much bigger. We're talking 52 families, around 395 known transporters in humans encoded by SLC genes.
Wow. And are they involved with drugs too?
Hugely. Many are drug targets themselves, or they play critical roles in drug absorption, getting drugs to where they need to go and getting rid of them.
Drug targets. Like you mean the drug is designed to affect the transporter?
Precisely. Think about antidepressants. Many SSRI, selective serotonin reuptake inhibitors, target SERT, the serotonin transporter.
And SERT is an SLC transporter.
Yes, it's in the SLC6 family. By blocking SERT, the drugs increase serotonin levels in the synapse. DET, the dopamine transporter, also in the SLC6 family, is another key target for different drugs.
Okay, this sets the stage nicely. We have these two major crews, the ATP-fueled ABC pumps and the larger, more varied SLC group doing facilitated or secondary active transport. Now, let's get into the core of it. How do these transporters actually impact our response to medications?
Right. This is where it gets really practical. We can think about it in two main ways: pharmacokinetics and pharmacodynamics.
Okay, familiar terms. Pharmacokinetics is
What the body does to the drug: absorption, distribution, metabolism, excretion, the drug's journey.
and pharmacodynamics.
Is what the drug does to the body, its effects, good or bad, usually at the target site.
Got it. Let's tackle pharmacokinetics first. How do transporters shape the drug's journey?
They're absolutely central. Many key transporters are located strategically in the cells lining your gut, your kidneys, and your liver.
The main entry and exit points and the processing hub.
Exactly. In the intestine, they influence how much drug gets absorbed into your bloodstream from a pill. In the liver and kidneys, they're crucial for pulling drugs out of the blood to be broken down or excreted in bile or urine.
So they're like selective gatekeepers for absorption and elimination.
Very selective. They often work hand in hand with drug metabolizing enzymes, forming this coordinated system for handling drugs.
Does this also affect where drugs go in the body? Distribution.
Yes, definitely. Transporters can mediate tissue-specific drug distribution. They can help concentrate a drug in the target tissue, which is good,
Or keep it out.
or keep it out. Think about PGP again at the blood-brain barrier. It acts as a protective shield, pumping many drugs out and preventing them from reaching the brain.
Right, the bouncer function. Okay, that's pharmacokinetics. Let's flip the coin to pharmacodynamics, what the drug does to the body.
Yeah.
You mentioned transporters can be drug targets.
Yes, this is a huge area. We already talked about CERT for SSRIs and DAT for drugs affecting dopamine.
Right.
Many other examples exist. Tricyclic antidepressants hit several neurotransmitter transporters. Drugs for ADHD often target dopamine and norepinephrine transporters. Some anti-seizure meds work via GABA transporters like J1.
So the transporter is the site of action for these drugs.
Correct. The drug binding to the transporter changes its function, which leads to the therapeutic effect. It's not just neurotransmitters either. Research is really heating up around targeting transporters involved in things like cholesterol, glucose, even nucleosides for cancer.
Are there recent examples of new drugs working this way?
Absolutely. There's a whole class for type 2 diabetes now, the SGLT2 inhibitors, drugs like empagliflozin, canagliflozin.
SGLT2. That's a transporter.
Yes, sodium glucose cotransporter 2, an SLC transporter in the kidney. These drugs block it, so less glucose gets reabsorbed back into the body, lowering blood sugar.
And they found other benefits too, right? For the heart.
Unexpectedly, yes.
Yeah.
Big benefits for heart failure and kidney disease, even in people without diabetes. It shows how profoundly these systems are integrated.
Any others?
There's lesinurad for gout, which targets URD1, another kidney transporter involved in uric acid reabsorption. And ivacaftor for cystic fibrosis, it actually helps the faulty CFTR transporter channel and ABC protein work better.
So targeting transporters is a really active area for drug development.
For sure, but transporters aren't always involved in positive outcomes. Let's talk about drug resistance.
Ah, yes.
Yeah.
Particularly in cancer, right?
Yeah.
How do transporters contribute?
It's a major mechanism. Cancer cells can become resistant in a couple of ways involving transporters. One is they can essentially shut down the transporters that normally bring the drug into the cell. Reduced influx.
So the drug can't even get in properly.
Exactly. Or, probably more common, they can ramp up production of the efflux pumps, the ABC transporters like PGP, MRPs, BCRP.
The ones that pump drugs out.
Right. So even if the drug gets in, it's immediately pumped back out before it can reach a high enough concentration to kill the cell. The cell basically builds better defenses.
Can we fight that? Like, block those pumps?
People are trying. Developing drugs to inhibit MDR1 PGP activity is a long-standing goal, though it's proven tricky to do effectively and safely in patients. But it's still an important strategy being explored.
Makes sense. Okay, what about adverse drug responses, side effects? How do transporters fit in there?
They're definitely implicated. Because they control drug exposure in cells and tissues, messing with transporter function can lead to trouble. We can think about it in maybe three main categories.
Okay, what's the first?
First, if transporters responsible for getting a drug into clearance organs like the liver or kidney, or getting it out via urine or bile, are less active, maybe due to genetics or another drug blocking them, the drug can build up in your bloodstream.
Higher systemic levels than expected.
Right. The classic example is the OATP1B1 transporter in the liver. It takes up statins. If your OATP1B1 is less active, statin levels in your blood can go up, increasing the risk of muscle toxicity.
I see. So problems at the main clearance depots. What's the second category?
Second, you can get the opposite problem at the target organ, or especially behind protective barriers. If uptake into a sensitive tissue is increased or efflux out is decreased, you can get too high a drug concentration locally.
Like the loperamide example at the blood-brain barrier.
Exactly. Loperamide, Imodium, usually doesn't cause opioid effects because PGP pumps it out of the brain. But if you block PGP with another drug, loperamide can get in and cause respiratory depression. Too much drug in the wrong place.
Okay. And the third category?
The third is when a drug interferes with the transport of the body's own natural molecules.
Like endogenous compounds.
Yes. For example, some drugs might block transporters that normally handle bile acids in the liver. This can disrupt bile flow and cause liver problems like cholestasis or jaundice.
You mentioned metformin earlier, the diabetes drug, and lactic acidosis.
Right. Metformin gets into liver cells via the OCT1 transporter. This uptake is key for its action, but might also contribute to that rare side effect, possibly by affecting energy metabolism. And inhibiting its efflux transporter, MATE1, could potentially worsen things.
So it's a complex interplay.
Very complex.
Right.
OATs and OCTs in the kidney are another example involved in eliminating many drugs, but can also contribute to kidney toxicity if things go wrong, or even affect how cancer drugs work on tumor cells if those cells express these transporters.
It really highlights the balance needing transporters for drugs to work, but also their potential role in resistance and side effects.
Okay, let's maybe zoom in a bit on the mechanisms. How do these transporters actually move stuff?
Well, first, it's useful to distinguish transporters from channels.
Okay. How are they different?
Channels are more like pores or tunnels. They open up and let specific ions usually flow through very quickly down their gradient. Think millions of ions per second.
Super fast. Transporters are different. They have to actually bind to their cargo, the molecule they're moving, then undergo a shape change, and then release the cargo on the other side. It's more involved.
So slower.
Much slower, maybe hundreds or thousands of molecules per second, not millions. They form this intermediate complex with the substance they're transporting.
Got it. Channels or tunnels, transporters are more like revolving doors or elevators that bind their passenger.
That's a good way to think about it. Now, within transport, we have passive and active. Passive first.
Sure. Passive means no energy needed.
Right. Driven by existing gradients, concentration, or electrical charge. Simple passive diffusion is just stuff dissolving through the membrane's fatty layer if it can. Facilitated diffusion still follows the gradient, but needs a transporter protein to help it cross.
Like the SLC facilitated transporters you mentioned.
Exactly. Still passive, just needs help.
And active transport needs energy.
Yes, to move things against their gradient. Uphill. Primary active transport uses energy directly, usually ATP hydrolysis.
Like the ABC transporters.
Like most ABC transporters, and also the famous sodium-potassium pump, they have their own engine.
Okay. And secondary active transport.
That uses energy indirectly. It couples the uphill movement of one substance to the downhill movement of another, usually an ion like sodium, using the gradient set up by primary pumps.
The hitching a ride mechanism.
Right. Can be symport, both moving the same direction, or antiport, moving opposite directions. Many SLC transporters work this way, like SGLT1, the sodium glucose transporter, is a symporter.
Okay, that clarifies the energy aspects.
Sure.
Now, what about how fast they work? Can they get overwhelmed?
Absolutely. Transporter-mediated transport is saturable, meaning there's a maximum speed of Vmax. It depends on how many transporter proteins are present in the membrane and how fast each one can cycle. Once all the transporters are busy working, adding more substance won't make the process go any faster.
Makes sense, like checkout lines at a store.
Exactly. And this relationship follows Michaelis-Menten kinetics, usually. There's also a kilema value, which reflects how tightly the transporter binds its cargo. Lower kilema means tighter binding.
Vmax is the speed limit. Kilem is how well it grabs the cargo. What about things that interfere? Inhibitors.
Right. Other molecules can inhibit transporter function competitively, where they bind to the same spot as the cargo.
Fighting for the same seat?
Yep. Or non-competitively, binding somewhere else but changing the transporter's shape so it works poorly. Or even uncompetitively, binding only when the cargo is already bound. Understanding inhibition and the kill value, which measures inhibitor potency, is crucial for predicting drug interactions.
Okay, this kinetic view is helpful. Now, physically, what do these things look like?
Yeah.
And how do they actually move things?
Structurally, both SLC and ABC transporters are proteins that snake back and forth across the cell membrane multiple times. Getting their exact 3D structures has been a huge breakthrough recently, thanks to techniques like X-ray crystallography and cryo-EM.
That helps understand the mechanism.
Immensely. For ABC transporters, we know they have these nucleotide binding domains, NBDs, on the inside.
Where the ATP binds.
Right. Binding ATP, breaking it down, hydrolysis, and releasing the products causes the NBDs and the membrane-spanning parts to change shape, effectively pushing the bound drug or substance out. It's often described as an ATP switch mechanism.
ATP binding opens it one way, ATP hydrolysis resets it, something like that.
That's the general idea for many efflux pumps. Binding and hydrolysis drive these conformational changes.
Okay. And the SLC transporters, their mechanism sounded more varied.
It is. But a common model for many SLCs is the alternating access mechanism.
Alternating access.
Imagine the transporter has a binding site for its cargo, but that site can only be open to one side of the membrane at a time, either facing outside or facing inside.
Okay.
Binding of the cargo on one side triggers a shape change, closing access on that side and opening it to the other side where the cargo is released. Then it flips back, sometimes called a rocker switch or gated pore mechanism.
So it rocks back and forth, exposing the binding site to alternating sides.
That's the core concept for many of them. It ensures transport is coupled to these conformational changes.
Fascinating mechanisms. Now, our bodies aren't just single cells. How does this work across layers of cells, like in the gut lining?
That's where vectorial transport comes in. Very important concept.
Vectorial, meaning directed.
Exactly. In polarized cells, cells that have distinct sides, like the apical side facing the gut lumen and the cisolateral side facing the blood transporters, are often strategically placed on one side versus the other.
Okay, why?
To drive directional movement across the whole cell layer. For absorption in the gut, you might have uptake transporters on the apical lumen side pulling the drug into the cell, and efflux transporters on the basolateral blood side pushing it out into circulation.
A coordinated handoff.
Precisely. Same idea in the liver, for moving things from blood to bile, or in the kidney from blood to urine. It requires this asymmetric localization of uptake and efflux transporters. The liver is a great example. OATPs on the blood side for uptake, MDR1, MRP2, BCRP on the bile side for efflux.
So it's a tightly orchestrated process across the cell. Now, does the body control how many of these transporters there are? Can it regulate them?
Oh, yes, absolutely. Transporter expression isn't fixed. One major way is transcriptional regulation, controlling how much mRNA is made from the transporter gene.
Like turning the production volume up or down.
Exactly. And this is often controlled by nuclear receptors. Proteins like PXR, CRR, FXR, they can sense the presence of drugs or certain body substances and then tell the cell to make more or sometimes less of specific transporters and metabolizing enzymes.
So the body can adapt its defenses or handling machinery?
It can. This induction or downregulation is a key mechanism behind some drug interactions and changes in drug response over time.
Are there other control mechanisms besides these nuclear receptors?
Yes, epigenetic control is also important. Things like DNA methylation, adding chemical tags to the DNA, can silence transporter genes in tissues where they aren't needed, or help keep them active where they are.
So like a longer-term programming of which transporters are made where?
Kind of. It adds another layer of regulation. We know methylation affects quite a few transporters, including OATs, OCTs, URT1, and several ABC transporters like MDR1 and BCRP.
Okay, so multiple levels of control. Now, we focus on ABC and SLC. Can we get a slightly broader overview of these two superfamilies in our genome? Let's start with SLC.
Sure. The SLC superfamily, as we said, is huge, 52 families, nearly 400 genes, incredible diversity in what they transport. Ions, amino acids, sugars, vitamins, neurotransmitters, metals, and, of course, drugs.
Mostly facilitated diffusion or secondary active transport?
Mostly, yes. And clinically, they're really important. Mutations cause many genetic diseases. Physiologically, they're essential for nutrient uptake and waste removal. Pharmacologically, they're key for drug absorption, distribution, elimination, drug interactions, and many are drug targets.
Any specific families that come up a lot with drugs?
The SLCO family encoding OATPs is crucial for statins, some diabetes drugs. The SLC22 family OATs and OCTs handles a vast number of drugs, especially in the kidney and liver. And genetic variations, polymorphisms in many SLC genes are linked to disease risk or altered drug response. Think SLC30A8 in diabetes or SLC22A45 and inflammatory bowel disease.
So a really diverse and critical group. What about the ABC superfamily overview?
Smaller group, 7 families, but also vital. At least 13 ABC genes are linked to genetic disorders when mutated. Functionally, many are involved in exporting drugs and toxins out of cells and tissues that protective role.
Like MDR1, MRP2, BCRP we keep mentioning.
Exactly. Those 3 are really key players in overall drug disposition, and their location tells you a lot about their function.
Where are they typically found?
Often on the exit side. Apical membrane of intestinal cells, pumping drugs back into the gut, kidney tubule brush border, pumping into urine, liver canaliculi membrane pumping into bile, and importantly, the luminal side, the blood side of barrier tissues like the blood-brain barrier, blood-placenta barrier, pumping things out of those protected spaces.
The gatekeepers pushing things out. You mentioned the MRP family within ABC.
Right, the ABCC family or MRPs, they often handle organic anions, particularly conjugates like molecules tagged with glutathione or glucuronic acid for excretion. MRP1 and 2 are examples. MRP3 is interesting. It's often on the blood side of liver cells, maybe handling overflow during liver issues. MRP4 deals with antiviral drugs and others. MRP5, nucleotide analogs. MRP6 is linked to a rare disease, but its exact cargo isn't fully clear.
And BCRP, ABCG2.
BCRP handles a wide range of stuff: neutral compounds, anions, chemotherapy drugs, statins, even dietary components. Genetic variants in BCRP are linked to gout risk and affect how some gout drugs are handled.
So knocking out these ABC transporters in lab animals must have big effects.
It does. Those knockout animals are often hypersensitive to drugs that are normally pumped out by these transporters, confirming their protective role. And clinically, MDR1's impact on digoxin levels and its role in drug interactions is very well established.
This really paints a picture of their importance. Let's focus now on pharmacokinetics again, specifically in the liver and kidney. How do transporters orchestrate drug handling there?
Okay, the liver, huge role. First, uptake from blood into liver cells, hepatocytes. This happens across the sinusoidal membrane, the blood-facing side.
And that's mainly SLC transporters.
Mainly. OATPs for organic anions, OCTs for cations, NTCP for bile salts. They pull drugs into the liver cell.
Okay, drug is inside the liver cell. Now what?
Now efflux. Across the canalicular membrane, the side facing the tiny bile ducts. Here, ABC transporters dominate. MRP2, MDR1, BCRP, BSFP for bile salts, MDR2 for phospholipids. They pump drugs, metabolites, and bile components out into the bile.
Using ATP energy.
Using ATP. This coordinated uptake, SLC, and efflux, ABC, creates that vectorial transport, moving drugs from blood clear across the liver cell into bile for elimination.
You had some specific drug examples for the liver. Statins.
Right. Statins need to get into the liver to work. OATP1B1 is a key uptake transporter. Once inside, they inhibit cholesterol synthesis. Efflux transporters like MRP2 are involved in getting them out into bile. OATP1B1 function is critical. Good uptake means high liver concentration, good for efficacy, and lower blood concentration, good for safety.
And the Gemfibrozil interaction messes with that uptake.
Exactly. Gemfibrozil blocks OATP1B1, reducing statin uptake into the liver, leading to higher blood levels and increased risk of muscle problems. A classic transporter DDI, drug interaction.
What about irinotecan, the cancer drug?
Irinotecan gets converted to active SN38. Both SN38 and its glucuronide metabolite are pumped into bile by MRP2. But in the gut, the glucuronide can get converted back to toxic SN38, causing severe diarrhea. Blocking MRP2 might actually reduce this toxicity by limiting biliary excretion initially.
Interesting counterintuitive idea. Any others quickly?
Bosentan relies on OATP1B13 for liver uptake. Temocaprolate uses OATPs and MRP2 for its dual liver-kidney elimination. Several blood pressure drugs like valsartan use OATP as MRP2. Even some diabetes drugs like repaglinide are influenced by OATP1B1 uptake, affecting how quickly they're cleared.
Clearly vital in the liver. Now the kidneys. Similar story of uptake and efflux.
Similar logic, yes. Primarily in the proximal tubule cells. For organic cations, positively charged drugs like metformin uptake from blood at the basolateral base membrane is mainly via OCT2 and OCT3.
Driven by electrochemical gradients.
Right. Then, efflux into the urine at the apical top membrane involves MATE1 and MATE2K, which exchange the drug for protons. OCTNs might also play a role here. Genetic variants in OCTs and mates affect metformin handling.
Okay, that's great cations. What about organic anions negatively charged drugs?
Also a two-step secretion. Basolateral uptake from blood, mainly by OAT1 and OAT3, often coupled to exchange with alpha-ketoglutarate.
Driving uptake against the gradient.
Yep. Then, apical efflux into urine is more complex, less understood, but likely involves MRP2, MRP4, possibly O8T4, though O8T4 might do reabsorption too, NPT1, maybe BCRP, and URAT1 is key here for reabsorbing urate back into the body. Again, genetic variants in OATs, ABCG2, affect anion handling.
Such intricate pathways for sorting and eliminating different kinds of molecules. Let's switch gears back to the brain. You mentioned transporters as drug targets there for pharmacodynamics.
Absolutely. The reuptake transporters, mostly SLC1 and SLC6 families, are critical for controlling neurotransmitter levels at the synapse. They recycle neurotransmitters like GABA, glutamate, norepinephrine, serotonin, dopamine.
And drugs hijack these.
Precisely. The SLC6 family is full of drug targets. JAT1 for tiagabine, anti-seizure.
Yeah.
NET for norepinephrine reuptake, hit by some antidepressants and cocaine. DATE for dopamine reuptake, target of cocaine, amphetamines, and CERT for serotonin, the target of SSRIs.
So manipulating these transporters is a major strategy in neuropsychiatric medicine.
It really is. And genetic variations in these transporters, like CERT, are linked to differences in behavior and susceptibility to disorders.
Now let's revisit the blood-brain barrier itself. From a drug perspective, what are the key transporter issues?
The BBB is formidable. Tight junctions between cells limit passive diffusion. But it's not just a wall. It's regulated by transporters.
Influx and efflux.
Both. SLC transporters bring in essential nutrients like glucose and amino acids. Some OATs might remove waste from the CSF, but crucially for drugs, the efflux pumps PGP, BCRP, MRP4 are highly expressed on the blood side.
Pumping drugs out as they try to enter the brain tissue.
Exactly. This is a huge barrier for getting drugs into the CNS. A drug needs to either be very lipid-soluble to sneak past or be recognized by an influx transporter, like L-DOPA using LAT1, and avoid being pumped out by the efflux transporters.
A tough combination.
Very tough. Many potential brain drugs fail because they just can't get past these efflux pumps in sufficient concentrations. Research is intense on ways to overcome this: nanoparticles, liposomes, blocking the pumps, designing drugs differently.
It's a major delivery challenge. Okay, let's move towards wrapping up with some broader concepts. What's the extended clearance concept, and how does modeling help?
The extended clearance concept just formally recognizes that liver clearance isn't just about metabolism breakdown. It's the interplay of uptake into the liver, potential backflow into blood, metabolism within the liver, and excretion into bile.
All the different processes happening.
Right, and often the initial uptake step by transporters like OATPs can be the rate-limiting factor for how quickly the drug is cleared from the blood overall.
So the transporter speed sets the pace. How does PBPK modeling fit in?
PBPK physiologically based pharmacokinetic modeling uses computers to simulate this whole process. It incorporates organ blood flows, tissue volumes, enzyme activities, and transporter activities.
Building a virtual body.
Essentially, yes. You can then use the model to ask, what if? What if this person's OATP1B1 activity is low due to genetics? What if they take another drug that inhibits MRP2? How will that affect drug levels in the blood versus in the liver?
So you can predict the impact of transporter variations.
Predict, or at least simulate, potential impacts. For instance, models suggest changing OATP1B1 might impact blood levels and maybe systemic toxicity more, while changing MRP2 might impact liver levels and efficacy liver toxicity more for certain drugs. It helps understand the differential impact.
Powerful tool. Now, we've mentioned genetic variations a lot. Can you summarize the clinical implications?
Well, major inherited defects in transporters definitely cause specific diseases, but the more common genetic variations, the polymorphisms or SNPs, are key drivers of variability in drug response pharmacogenetics.
Real-world impact.
Huge impact. The SLC01B1 variants affecting statin levels and toxicity risk is a prime example now often tested for. SLC22A variants affecting metformin response, ABCG2 variants influencing responses to drugs like rosuvastatin or allopurinol, variants in MRPs linked to toxicity of drugs like tenofovir. It's becoming increasingly clear that an individual's transporter genetics can significantly influence outcomes.
Which leads to the idea of personalized medicine.
Exactly. Tailoring drug choice or dose based on transporter genotype is already happening to some extent and is likely to grow.
And finally, how do regulatory agencies like the FDA view transporters now?
They view them as critical. Because transporters are major determinants of drug disposition and variability and a key source of drug-drug interactions, agencies now mandate investigating potential transporter interactions during drug development.
So drug companies have to test for this.
Yes. There's specific guidance recommending in vitro testing against a panel of key transporters, OATP1B1, OATP1B3, PGP, BCRP, OCT2, MODIPORT, OAT1, OAT3. If potential interactions are seen in the lab, clinical DDI studies might be required.
Why those specific ones?
Because they're known to be clinically relevant, involved in significant drug interactions, or impacting pharmacokinetics of many drugs. The gemfibrozil-cerevastatin interaction, which led to fatalities and market withdrawal, really underscored the importance of understanding transporter DDIs for safety.
So a huge shift towards recognizing their importance in drug safety and efficacy.
Definitely. Identifying which transporters handle a drug and what might interact with them is now a standard part of drug development and labeling.
Okay, this has been incredibly comprehensive. Let's try to summarize the key takeaways.
I'd say membrane transporters are essential proteins controlling drug movement everywhere in the body.
Right. Influencing absorption, distribution, effectiveness, side effects, the whole picture.
And your individual genetic makeup for these transporters can significantly alter your personal response to medications.
And understanding them is now absolutely vital for developing safer, more effective drugs.
Couldn't have said it better.
It really is remarkable. Which leaves me with a final thought to ponder. We've seen how intricate this transporter network is and how much it influences drug action. Could we, down the line, see medicine tailored not just to our general genetics, but hyper-personalized to the specific activity and variants of our individual membrane transporters?
That's the dream, isn't it? Truly individualized pharmacotherapy based on a deep understanding of how your body's transport systems work. It's complex, but maybe not impossible.
Something for us all to think about. This has been a fascinating deep dive. If you found this interesting, we really encourage you to maybe look into transporters related to any medications you take or conditions you have.
Yeah, having that knowledge can empower really good conversations with your doctor or pharmacist.
Absolutely. This is just scratching the surface, of course. There's so much more to learn about these vital molecular gatekeepers.